Okay, welcome back to Animal Cognition, and this week we're going to be talking about timing and number. Both of these concepts are often talked about together because they both have to do with the perception or the processing of magnitudes, right? So amounts. So we're going to start off today with this question: how do animals keep track of time? I'm sure we all have some experience with pets. Always waking us up at the same time of day for their food, right? So we know inherently that it seems that our pets, at least, have this kind of sense of time. But even animals that aren't our pets, even animals that are not domesticated, they still have an intrinsic perception of time. They can keep track of time. There are two main types of timing, or two ways we can break down timing. The first is known as periodic timing. So this is for a very long period of time. This has to do with a 24-hour period or the day. So there's cyclical activity here, right? So your dog is waking you up at the same time every morning, right? It's a cycle, day-night cycle. There's also a much shorter period of timing, known as interval timing, and this has to do with short durations of seconds or minutes. And we'll get into some examples for that in a little bit. Okay, so let's start out with talking about periodic timing, that kind of day-night cycle. So we might think of this as our circadian rhythm, right? Might be familiar with that, also known as our internal clock. And basically, just about every organism, that includes non-animals as well, almost every organism that has been studied shows some kind of rhythm of activity or state that matches the day-night cycle of Earth, right? It's very important. It's a very big cue. We all evolved on the Earth, right? So it makes sense that we are attuned to it and have these rhythms. So the rhythm, interestingly, our circadian rhythm, can persist even when we don't have light or dark cues available to us. So if you put a person in a room without access to the sun or cues about what time of day it is at all, so no clocks, they'll still have kind of a general rhythm of knowing what time of day it is. Okay, so our circadian rhythms can undergo something called entrainment. So it can fall into synchrony with the day-night cycle. So our circadian rhythm that's inherent isn't exactly 24 hours long. It's basically, it's also known as a free-running rhythm. It's close to 24 hours. It's something like 23 and a half hours. So if you put that person in a room without any outside cues, they're not going to know exactly what time of day it is, but they're going to be very close. So that's our free-running rhythm. But as soon as you give someone those cues of what time of day it is, it undergoes entrainment and falls back in line with the external day-night cycle. So animals' circadian rhythm is usually discussed not in terms of cognition, but more as a physiological process. But we do know that animals can learn about events that occur at certain times of day. So going back to that feeding your dog or your cat example, so they can link those events to certain times of day, and that can link the event to an internal clock. In one experiment, the researchers had garden warblers in this kind of enclosure. Okay, so in the middle, what you're looking at is the general enclosure where the birds were housed, and then surrounding that enclosure there are 4 separate feeding rooms. Now each of the feeding rooms were closed at certain times of day, and then one of the enclosures was open. So basically, for different blocks of time, so 3-hour blocks of time during a 12-hour light cycle, right? So the birds are feeding in the daytime and sleeping at night. During the 12 hours of daylight that the birds are given, there are 4 blocks of 3 hours for each feeding room, okay, that are accessible, that are open. So what ended up happening is that the garden warblers linked specific rooms to different times. Even though after a certain amount of training sessions, all of the rooms were available to feed, the birds still fed in the particular order that they had learned. Okay, and we can see those results here. So what you're looking at on the x-axis is the number of choices made by the birds, so where they're trying to go feed. And then on the y-axis, we have time of day. On the right-hand side, you can see the different room numbers labeled, so 1, 2, 3, 4. And then if you look at the bar graph for 1, for example, you see these lines above the bars, and this is showing the period of time that the rooms were open or accessible, right? So you can see during training, the birds are learning where they have to go at each time of day to feed. And then during testing, even though all rooms were accessible, the birds are actually going to the areas that they learned much more frequently than when they were training, right? So they've actually created this association very well. They know that in the morning they have to go to room 1, and at the end of the day they have to go to room 4, right? Even though at this point all rooms are accessible to attend. And you can see that there are some responses here and there to those other rooms, but most of the responses are associated with a particular time of day. Now you might be thinking, what if the birds are just learning a pattern? Go in room 1, then room 2, etc. So they did a follow-up to this experiment where birds were prohibited entry into any of the rooms during one 3-hour block. Okay? And then when entry was again permitted during another block, the birds entered the appropriate room for that block. Okay, so does that make sense? Basically, on some trials, they'd prohibit the birds from entering in one room, and then on other trials, they'd allow them back in. The birds still reliably entered the appropriate room, and that showed that performance wasn't just based on a pattern, but linking food locations, so food and a place, to the circadian rhythm. This also inherently makes sense, right? If you think about an animal needing to forage for food, it makes sense to know when certain food resources are going to be available. We all know that saying, the early bird catches the worm, right? Certain food resources are probably available at different times of day. So it's important to know where to go at what time in your and associating it with your circadian cycle. So it's not super shocking that animals can remember times of day and pair them with locations where food is available. Okay, so what is the mechanism for this time and place learning? Well, for the garden warblers in the previous example, it's likely that the state of the garden warbler's internal circadian clock was acting as a contextual cue, a contextual stimulus to then associate the food. Again, the example of feeding our pets comes up. Our dogs or our cats are associating the time of day as a contextual cue with when they get to eat, right? An important piece of information when you get to eat. We also know from that garden warbler study that the circadian clock in the garden warblers was likely entrained to the light-dark cycle, right? So they had 12 hours of light on, 12 hours of lights off. So the people who did this experiment actually did another follow-up where they had the lights on the whole day, for a whole 24 hours. And birds still visited the rooms during the specific 3-hour blocks that they were supposed to at the right times. But this really only lasted for the first day of testing. And after that, the visits to the different rooms started to diverge from the 24-hour clock, right? So it kind of started to follow this free running rhythm. So you'd see things sort of shifted a little bit over time. So in human beings and in animals, a neural mechanism we know about is the suprachiasmatic nucleus, or the SCN. So mammals all have a suprachiasmatic nucleus, and it's found in the hypothalamus, right? So basically, what happens is that the eyes provide information to the SCN about levels of light, okay? And then that's sent from retinal ganglion cells, so from your retina, to the SCN. The SCN then passes this information to the pineal gland, which is important in the secretion of the hormone melatonin. So the presence of light can block the release of melatonin, which can, of course, disrupt sleep. This is often why we're told not to look at our phones before we go to sleep, right? Because that's giving our retina light information, which is then sending information to our suprachiasmatic nucleus, which is blocking the release of melatonin in our pineal gland. Okay, so that was a lot of information. But basically, what I'm trying to say is that in mammals, the light entrained circadian clock is thought to be located in the suprachiasmatic nucleus. So this is a very important structure for our circadian clocks. And some other animals actually also have non-retinal photoreceptors. So what does that mean? That means that they have photoreceptors, so receptors for detecting light, that are found not in their eyes. So they'll, of course, have photoreceptors in their eyes, but quite a few different species, so there's evidence in birds and amphibians and in some fish, they'll have photoreceptors in their brain. So light can directly be a stimulus for certain structures related to timing in the brain. So that's I find that an interesting little piece of information, that not all photoreceptors important for timing are all found in the retina. In some species, they're also found just in the brain. So light just hitting the brain can cause issues with circadian clocks in different species. Kind of cool. Okay, and here's just a nice little diagram of showing that process that I just talked about. So light's being picked up by the eye, activating the suprachiasmatic nucleus, sending messages to the pineal gland, which then has implications in how melatonin is released, right? And so in some animals, they'll also get direct stimulation right into the brain. Now, not all animals have a suprachiasmatic nucleus. Like I said, mammals do. Birds certainly do, although their system's a little different than this. Some fish, I don't believe, have a suprachiasmatic nucleus, but they certainly do have a pineal gland, and they'll have other associated structures that are similar and important in timing and in the circadian clock. All right, so we talked a bit about periodic timing. Now let's talk about timing intervals. So these are shorter periods of time. So we already kind of touched on this idea when we were talking about associative processes, right? So associations between two stimuli are formed more easily or more strongly when they occur close together in time, right? Here we're talking about the ability to time short durations and discriminate between them. And these kinds of stimuli that govern interval timing can occur at any time of day. So that's contrasting to periodic timing, right, where the time of day is quite important. Here it can occur at any time of day, it's shorter, And associative learning is related, but here what we're focusing on is the ability to time short durations and discriminate between them. So a little bit different. An important procedure for examining this kind of timing in animals is something called peak procedure. And this work was done by Roberts, who did a lot of his work here at Western, actually. So what happened here is that rats basically were exposed to a light, okay, so they were given a stimulus that was a light, and it signaled that food could be obtained by pressing a lever 20 seconds later. So a light comes on, and in 20 seconds, you have to press a lever to get a food reward. Now, on some of these trials, no food rewards were given, so these are known as empty trials, and these are the important trials here in this procedure. Okay, so on these empty trials, the rats, of course, are pressing the bar multiple times. They're not just pressing once exactly at 20 seconds. So what happens is that the rats' responses, so they're pressing the bars, right? It peaks very close to 20 seconds, actually, and follows a relatively normal distribution. So basically, you can see on this graph that the rate of responding is highest, closest to 20 seconds, and then kind of tapers off. Similarly, if you look at a stimulus signaling food to be reward after 40 seconds, instead, also shown on the figure, you see responses peaking again at 40 seconds and following a normal distribution. But the spread of the curve is greater. See how it takes on a wider range, right? So the variability of the curves is actually proportional to the length of the interval being timed. Okay, so the shorter the interval is and the longer the interval is, you'll see this similar pattern. So this has been termed as something known as the scalar property. And this might actually be kind of familiar, right? Because what's happening here is actually fitting in quite well with Weber's law, right? And we see that other experiments kind of show similar results. So most of our work in interval timing comes from rats and pigeons. Pigeons are, of course, also often seen as the lab rat of the avian world. But we see this kind of interval timing across many vertebrates, but also invertebrates too. So some work from Boisvert and Sherry, and Sherry here is the same Dave Sherry that did that black-capped chickadee work that was really important. So David Sherry was a professor at Western for many, many years, so some more work from Western in this area. But what they did was they tested these fixed interval procedures with bumblebees. So they had to change the way that their apparatus would look a little bit, because you have to, right, adapt apparatuses to the species that you're working with. So here, in this case, the response in question would be the extension of the proboscis, which is something that naturally happens during feeding. So the proboscis in a bumblebee is basically their tongue that they use to suck up honey or nectar. To suck up nectar, not honey. They make honey later, right? So here, a bee could obtain sucrose, so there's a sucrose reward, by extending its proboscis through a small hole in the experimental chamber, which you can see designed there. And they find basically similar patterns of results that you would expect to find in invertebrates, in pigeons, and in rats. So we have evidence of interval timing in rats, in pigeons, but in bumblebees as well. Okay, so we've talked a little bit about timing in the lab and how to study it, but what about timing in the wild? So we've discussed this already a little bit, that it might be advantageous to be able to keep track of time to know about the location of food sources, right? when they are, when is the best time to eat from these food sources, right? And when we look at hummingbirds, or pollinators, we have a great example of this. So hummingbirds and bumblebees are two examples of pollinators. They both feed on nectar, and in that process, they carry pollen around with them, and they're pollinating other plants, right? So when it comes to pollinators, these species often show something called resource fidelity. So they repeatedly visit the same nectar sources. They go back to the same flowers. And flowers do replenish their nectar after a pollinator has fed, but this process takes time. So feeding efficiently requires pollinators to have schedules, right? They have to visit food sources after a certain amount of time has passed. So this would require remembering not only where the feeding site was, but how much time has passed since the last visit. So in an experiment with rufous hummingbirds, experimenters had an array of 8 different flowers. Artificial flowers, right? So they're filling them up with sucrose, but they have a color and a shape to them. And you can see what the artificial flowers looks like there in the image. Basically, 4 of the flowers were refilled every 10 minutes, and the other 4 flowers were refilled every 20 minutes. Now, what ended up happening was after several days, birds learned this schedule. So again, this is happening out in the wild, remember, so they're observing the behavior from different birds. So here we're looking at the behavior of just one bird. It shows 3 different birds in your textbook, but all of the results are generally the same. I hope you can't hear my cat. It's okay if you can. Okay, so if we look at the results, we can see the results for the 10-minute flower and the 20 minutes flower. The 10 minutes is in gray and the 20 minutes is in white. So what you see is that most of the responding, right, for the 10 minutes flower happens in the 10-minute range. And most of the responding for the 20 minutes flower happens in the 20-minute range. So basically the hummingbirds learned that the time until refilling differed among the flowers, right? They're visiting the 10 minutes flower before the 20 minutes flower. And the rate of responding is similar to what we see in the lab. Now there's a lot more variance and it's not exactly the same, but we're in the wild here, right? Okay, so now we're moving on to asking, how do animals and humans time intervals without a watch? We talked about some of the mechanisms with the suprachiasmatic nucleus for circadian timing, but how do we do this for shorter time intervals, and how do we do it without clocks? Currently, there are 3 general models of how we interval time, of how animals and humans interval time. The information processing model, the oscillator model, and behavioral models, which we'll go through each one next. Okay, so the first model, the information processing model of timing, has a few different names, sometimes known as the pacemaker or accumulator model, is basically a model where comparison to a representation in our memory is important. So that comparison piece is pretty important. But what it relies on is a hypothetical pacemaker. Okay, so think about a hypothetical metronome that you have somewhere in your brain. Okay, so it emits pulses at a constant rate. Metronome, tick, tick, tick. So when a to-be timed event starts, a hypothetical switch is activated. Basically, you're turning on that metronome. Then the pulses accumulate until the event ends. So you have that hypothetical metronome, tick, 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 until your event that you have to time ends. And then the information held in your accumulator, so how many pulses you accumulated, is held briefly in working memory. Then the contents of working memory are transferred to reference memory, which also contains information about perhaps in the case of a lot of the examples we're talking about, it could contain information about whether a response was rewarded or not, but it can be applied in other contexts as well, right? But basically the important part with memory there is that the working memory of the magnitude is then compared to reference memory to then guide a decision process. So you can see that kind of visualized here. We have a signal that starts the clock process. So we have our pacemaker, a switch to turn the pacemaker on, to turn the accumulator on, and then once the event that we have to time ends, we take that accumulated information into our working memory and compare it to our reference memory, right? And that can give us crucial information with decision making. Here it's just shown as a simple yes or no, but of course this could be more complicated. This is just a model of how this might happen. So while the information processing model is useful, it doesn't really match onto exactly the processes of timing neurobiologically. So it doesn't really fit the neurobiology of timing super well. So the oscillator model was developed to kind of fit with the neurobiology of timing a bit better. So here, instead, they don't have just a pacemaker. The pacemaker is replaced with multiple oscillators. In other words, this model posits that there is many oscillators. They're mechanisms that fluctuate, and they're in a rhythmic sinusoidal matter. And this maps more on to our biology, right? So there's not just a pacemaker that's emitting pulses. It's not a metronome, but there's multiple oscillators, things that fluctuate in a rhythmic manner. So we can see what that might kind of look like here. Multiple different oscillators, they have different rates of change, but they're all kind of in this sinusoidal pattern. Now, how the oscillator model actually works can get a little mathematically complicated, so we're not going to go in too much detail about it, but knowing it at this level is perfectly sufficient. So the other important thing to just take away from the oscillator model is that it is more accurate to fit our underlying biology. An interesting question to lead into our final potential mechanism that we're going to talk about is, do we always need a clock, internal or external, to time? So some explanations of interval timing don't incorporate a pacemaker or an oscillator or any other clock type of mechanism, and instead account for timing in other ways. And these are behavioral models. So these do not rely on any kind of internal clock, but instead describe timing in a more traditional behavioristic manner, rather than through a kind of cognitive model like the ones we just talked about. So here the key is that the behaviors that animals engage in actually help them indicate duration. So the behavioral state at the time of reinforcement can come to be associated with rewards. Another potential idea here in these behavioral models is something known as memory decay. So in lab situations where animals receive food at regular intervals, it is possible that the decay of memory from one reward to the next can itself be associated with food. Okay, so for example, if a pigeon receives a reward, pecking every 15 seconds or so, any one event of a reward creates a memory trace. So this memory becomes weaker or decays over time, right? And its strength at 15 seconds might actually be associated with the reappearance of food if the rate of decay is similar, trial after trial. So in other words, duration is indicated through the strength of a memory.